Hi, everyone. I'm going to be talking about this paper titled Computational Optimization of Antibody Humanness and Stability by Systematic Energy-Based Ranking. This paper comes out of the Fleishman lab at Weizmann Institute of Science. Some background on antibodies. They have the binding region in the FV or variable region. The CDR loops on the tips of the FV mediate the binding, and they're resting on this framework region, which is determined in vivo by 3 different genes on the heavy chain, V, D, and J, on the light chain, V and J. So typically antibodies are developed from mouse antibodies and need to be humanized in order to limit immunogenicity. Typically this is done using homology modeling. However, these authors use combining V and J genes to create 20,000 plus human acceptor frameworks instead of homology modeling, and then screen all of these different frameworks using Rosetta. So they graft on the parental CDRs, which mediate the binding, and then use their newfound frameworks. They did the screening using SCFBs of these grafted CDRs on their frameworks using Rosetta Energy. So first they take their parental antibody sequence and their human acceptor frameworks determined by the V and J gene combinations. They graft on those CDRs. And then the structure energy is ranked and clustered by V gene using the Rosetta All-Atom Energy Function 2015. So the goal is to be able to screen for humanized antibodies while maintaining parental affinity and stability. They used wet lab validation to prove that they did this, and that it's more efficient than homology methods, which typically require some back mutations after humanizing. So they showed that compared to their parental antibody, their computationally humanized antibody was more human, achieving acceptable humanness levels. They also showed that their humanized antibody was more stable using differential scanning fluorimetry. They showed that they had acceptable and even improved binding kinetics. They used a polyspecificity particle assay to show that their humanized antibodies were within an acceptable range of polyspecificity. And then they used a CSSINS self-aggregation assay to show that their antibodies were within an acceptable range. And this correlates with low risk of high viscosity and obsolescence, which are critical for therapeutic development. So in conclusion, their humanizing process using V and J gene recombination and then energetic and structural rankings for screening all of those recombinations was more efficient than homology-based methods because they could do one-shot humanization with no back mutating.